Okay, it is 2 p.m. now here, so let's make a start. Welcome everybody, and thank you for joining us today. I'm Karina from SynBio.Oxford. We are a student-led society based at the University of Oxford, which aims to promote synthetic biology by bringing you talks by experts in the field. So today's event will revolve all around the potential of SynBio to improve existing technologies and provide new ones for therapeutics and diagnostics. It will be split into 3 parts. So first off, we'll have a discussion with academics with diverse specializations in these fields. That will be followed by presentations by 5 startups that we've invited to present to you their approach to improving healthcare. And finally, we'll have a panel discussion with investors in the space. We will welcome your questions throughout the event. Please put them in the Q&A box, and the respective chair of the session will pick them up. So before we start, I just want to thank the incredible SynBio.Oxford team for putting this event together. Also thank you to our sponsor, Integrated DNA Technologies, or better known as IDT. And thank you so much for all the speakers who are volunteering their time for this event today. I hope everyone who's attending will enjoy it, will learn something new, or see things in a different light. So without further ado, I will hand over to the chair of the academic panel, Dr. Harrison Steele.
Thank you very much, Karina. Welcome everybody. I hope you've had a good Tuesday afternoon so far, and thank you all for joining us. So we have an excellent group today of transatlantic synthetic biology researchers from focuses in therapeutics and medical applications of SynBio. We have Dr. Wong from Boston University. We have Dr. Elise Kesha from University of Edinburgh. We have Dr. Pardee from University of Toronto. And finally, Dr. Francesca Ceroni from Imperial. I won't say any more about them because they now have a couple of minutes each to kind of introduce themselves, say a little bit about what they do and what they're all about. So I don't want to steal their limelight. So without any further ado, I'll hand over to Wilson, if you'd like to take us away.
Yes, hi, thank you. First, I want to thank the organizer for the invitation. So looking forward to it. So yeah, my name is Wilson Wong. I'm associate professor from Boston University. My work is focused on mammalian synthetic biology with emphasis on T-cell engineering for therapeutic applications, for cancer, but we also are very interested in autoimmune diseases, transplant rejections, and also diagnostics as well. So basically, it's a medical application of synthetic biology.
Great, thank you, Wilson. Now, Elise, if you'd take us away.
Yeah, hi. So my name is Elise Kesha, and I am a group leader and lecturer at the University of Edinburgh in the UK Centre for Mammalian Synthetic Biology. So in my group, we're interested in engineering synthetic communication or signaling systems in mammalian cells, and really in the hope to interrogate various cell-cell interactions. So we're particularly interested in we're using a lot of synthetic receptors of different architectures, trying to detect interactions between, for example, first mammalian cells. So we have a project, Sofia Semenyuk in the lab is a PhD student who's looking at interactions between macrophages and cancer cells, hoping to interrogate those interactions in animal models of metastatic cancer. We're also interested in looking at interactions between mammalian cells and bacteria, and in particular, gut cells and the microbiome. And also we're building receptors to detect various metabolite extra or intracellularly in mammalian cells. And really our first aim is to first develop those in the lab in vitro, and hope that we get systems that are robust enough to then move into in vivo animal model work.
Great, thank you, Elise. And over to Keith.
Good afternoon. I thought I'd start out giving sort of a little background. It was my road to synthetic biology was not a straight line. I have sort of fundamental training in science with 2 degrees in botany, and then my PhD was in protein structure and function. And then for my postdoc, I decided to sort of take a curve towards applied biology. I've always been interested in building things, and actually was a carpenter for 3 years. And so I did my postdoc with Jim Collins at the Wyss Institute, which sort of transitioned my fundamental science background into applied areas. And now our lab at the University of Toronto is focused on primarily on cell-free synthetic biology. And we use that to provide a sort of like a abiotic, biosafe format for taking tools out of the lab and into the real world. And we use this to run diagnostics, as well as starting to sort of work in the area of distributed manufacturing for therapeutics. And so for the diagnostics, we've built sensors for things like Zika, chikungunya, and of course now SARS-CoV-2. And we've performed field trials, patient trials in Latin America, and those are ongoing. And another feature, I guess, of our work is because it's self-free, we have an opportunity to bring hardware into direct contact with engineered circuits. And so we've done quite a bit of that, and that's a theme. And I guess to fit sort of the theme of the day, we've got 3 trainee-led startups that have come out of the lab that are very still early days, but very proud of them. And so that's it. Looking forward to it. Thank you.
Thank you, Keith. And over to Francesca to round it up.
Yeah, good afternoon, everyone. So first of all, thank you for inviting me. I have the impression I am really among giants in the field, so I'm really honored to be here. And basically, I'm a lecturer in the Department of Chemical Engineering at Imperial College. And the track record of the lab is on looking at host construct interactions in bacteria. So looking at how the synthetic constructs that we build impact the host physiology, and how to identify, design rules to make these constructs more robust and best performing over time. And starting from this background research, we are now, so the lab is quite new. We are starting looking at this topic in mammalian cells, so host construct interactions in mammalian cells for bioproduction and eventually therapeutic applications. And we are also starting projects on the foundational tool side, because we are interested in the interaction between tumor cells and immune cells in the tumor microenvironment. So we are looking at developing cell-based and also synthetic cell-based tools for SynBio. Yeah.
Thank you very much, Francesca. Well, that is great introductions from everybody. So I should say now we'll go to a kind of question answer discussion session. So we have some questions prepared ourselves, but also for everybody in the audience, feel free to put any questions you have into the Q&A box, and then we'll get through them in turn. Hopefully get around to answering them all. But to begin with, so one thing that many people have been interested about and raised beforehand was thinking about the main obstacles to going from our research in SynBio in the labs, we're all at universities mostly, to actually getting into applications into the hands of a clinician or into the hands of a user. And this is obviously a challenging process. So I'm really interested to see what everyone has to say about the balance academia currently has between perhaps the blue sky research and the more applied going into the clinic research. We've heard from several, like Francesca was just saying that she does a bit of both. Keith said there's been 3 student startups from his own lab, for example. How do you think we can balance these things to make sure, on one hand, we are doing the fundamental science, which is required, but also making sure it actually gets to the people that need it in the end? Does anyone want to kick us off on that?
Yeah, I can. Sorry, Keith, you want to go?
No, go ahead, Wilson, please.
Yeah, I face this problem all the time. So, as an engineer, I guess the main thing is that, I would say, I try to listen to like clinicians and investors and people in the company, say, okay, what do they want? Sometimes they see, the work coming out of our lab, and they start coming to us and talk to us, but they would be like, well, I think one of my challenges is that, it's about robustness, right? How well they perform, right? That's what will happen a lot of time in the lab, is that you do something, you try to publish in a really fancy journal, and then you try to get very sophisticated so they look so cool, right? And then it barely works, so you publish it, and then and then the smart investor, the clinician, they smelled it, right? They like, okay, this doesn't look like it will work in patient very well. But then they start telling you what the challenges. So what I do a lot of the time, I always say, okay, there are two sides of me. One is I want to see what I can do with synthetic biology. So this is where I push the most fancy stuff and see where the kind of the lay of the land, what's possible. But then at the same time, I also listen to clinicians and investors and people who start companies say, like, here's what we need. And then I start trying to see from what we can do, is there something that we can, like, a little bit more robust, a little bit simpler, that I can translate more readily into the clinic. So it's both, right? Because as an academic, that's what we allow to do. That's what we're supposed to do, is bridge this landscape from like very fundamental, very pie in the sky discovery, and then find ways to bridge that, right? And I feel like this is really our job in the academic to do that, right? In the clinics and in companies, they don't have that luxury to do this kind of work. So it's really up to us.
Yeah, I can, or Francesca, did you have a?
No,
Oh, okay.
I can go after you. Can you imagine? Of course.
Okay, sure. So I agree with Wilson. I think also in the work we do, which is diagnostics, the line between what we're doing and what is needed is a little more like obvious, because we're doing diagnostics, for the past year, it was fairly obvious we needed to build what we needed to build. And I guess I would add also, one of the things we try to do in our lab is as sort of pushing the envelope as far as we can on the technology development side, but then also sort of be somewhat vertically integrated so that we actually take those technologies out and try them in the field. And that gives us kind of this middle ground where we can make sure they're robust and we get to sort of firsthand get a feel for things that aren't working, right? What are the problems when you actually try to move the technology that, forward. And like Wilson, we're molecular biologists. And so we need to surround ourselves with experts, right? So we have at the team sort of like pathologists that are, or virologists who have expertise in the pathogen of interest, and also clinicians who have expertise in the implementation of these things. And so, yeah, I would say, that's another key thing here is, right, to surround yourself with all the sort of knowledge users that would end up using it.
I wanted just to echo what Keith was saying, in the sense that for us, for example, one thing is the diversity of the hosts that we're using in respect to what is the actual end up, final host where things should be working. So the robustness or the functionality. You may test them in the lab, and that is one thing, because you use, but we use cell lines that are not really any way close to what then the clinicians use. So that is one thing, for example. So as Keith was saying, having a network of people like clinicians that can provide you with the proper host, but then also the survival of this host, the maintenance of these primary cells and whatever is not that straightforward. And the other thing is, for example, if I think about the tumor microenvironment and the analysis of this kind of interactions, of course, having the right models, like systems where you can actually test your engineered cells is not that straightforward. So there are some, drawbacks or weird behaviors that you can encounter. So it's not that, for us at least, it's not that easy.
So I feel we have a slightly different perspective. It's early days for us in the lab. And also the type of application we are looking at is really building tools for biomedical research at the moment. So we are quite far from translational application yet, but already we can see that there is a big step between our in vitro testing and in vivo function. And so we have a lot of technical hurdles that come with the fact that we're working with mammalian cells that don't always behave as expected or as you would expect from whatever tools been published. And so we require a lot of optimization, and we feel that there is very little of this optimization that can be done transiently, so with transient transfection, for example. So we spend a lot of time generating stable cell lines and then testing them, and that limits a lot our capacity to test various conditions or test different architectures of the different systems. So for us, really, at the stage we're at, the main obstacles are technical obstacles.
Yeah, that's a very good point. I'm interested in other people's thoughts on how, you know, I mean, you're describing this challenge of really the low-level technical work is just still so difficult. Whereas, for example, I mean, some academics are also focusing a lot on the translation, and we're also kind of pushed into that, say, by journals, by funding bodies. They want to see somebody who's doing everything from the low level all the way through to, you know, shipping it into somebody's hand, which I mean, it would be nice, but obviously it also requires a massive skill set, and everything takes 5 times as long. Do you think we are trying to be jacks of too many trades, and maybe we should, you know, pick one extreme and just really narrow in there? Or do you think it really adds a lot to have individuals covering the whole spectrum? I don't know, what do you think, Elise? Do you have aspirations to become more and more clinical, or are you really kind of focused on?
I've got aspirations of linking with clinical people. I think I'm best at working out those technical hurdles for now. And this sort of jump into translational applications, I feel is maybe best handled as collaborations rather than me being this jack of all trades. That's my opinion.
What does everyone else think? What do you think these major obstacles between research and clinic are that we could maybe think of new ways to overcome rather than just butting our heads into the wall alone?
If I can very quickly echo Elise, like I think the stable like engineering of mammalian cells is still for us is still a challenge, because I mean the silencing comes every time, and even if there are landing parts that you can use for you know multiple reintegrations, actually they're not always very stable. So we are facing the same problem, Elise. If that can make you feel better, I mean it's not straightforward at all, and I guess that not always in literature the actual stability of this kind of issues are covered.
Yeah,
so.
I agree that that's probably how to manage your expectations. So I think so as I said, we work with different synthetic receptors, and from the way they've been published, they have been portrayed as fairly plug and play type systems, but it feels to us that in mammalian cells nothing is plug and play, and each time you try something slightly different, you start from scratch again. So we have also the silencing, and also the fact that you need to incorporate so many things together that and co-transfection there's so much you can do with co-transfection that it's limiting a lot in terms of time and scope what we can do.
Keith, how do you feel about this? If we pick up a paper-based biosensor from your work, is it going to work first time every time, or is it also subject to these same challenges of mammalian cells?
I mean, absolutely, there are challenges, right? So I think we have maybe a little easier maybe sandbox, because we're doing hardware, which, you know, we can literally order the part and it does what it says it's going to do. And in the case of our sensors, they're in vitro. So there's much fewer sort of like, you know, things fighting us, right? So like we don't have a cell fighting us. But, you know, absolutely, you know, when it comes to practical implementation of diagnostics, you know, how do you prepare the sample in a low resource setting from a patient? How do you ensure reproducibility? All of these things, absolutely, when it comes to sort of like actually doing it, it's challenging, right? But I would say for us, it's something that seems to be working, and it's sort of the pull from the market need or the patient need is helping us decide what to work on.
That's an interesting point. I think that was brought up by Wilson, right? When you started talking about the an approach of going really for the fancy frontier versus also just talking to try and solve the simpler problems, but still challenging ones. How do you balance between those two? It must be very difficult to predict ahead of time what really is going to be the fancy frontier and what really is a safe bet.
Yeah, I haven't figured that out. So still a lot of trial and error. So I just want to echo what everybody said, right? I think about it, you know, a little bit. It's the same thing, but a little differently in terms of it's basically, you think about engineering, right? Usually you understand your substrate that you're working with very well, right? You're working with a piece of metal, piece of wood, you know very well what the properties are, or only a few properties that you care about, right? Whereas with mammalian cells, with T cells, you don't really know what that substrate you're working on. I think that's the hardest thing for us. So because of that, you try to do something really, really simple, or you want to be very robust. It end up switches, right? We work with CAR-T, for example. That's even that. It's kind of like a switch. And you know, and then you just hope that it doesn't break too much, too many things while you put them in there. And then all these other fancy circuits, I try, and then you know, I just talked to Michael Elowitz last week. And I asked him exactly the same question because he studied a lot of genetic circuits, so he's one of the best in the world. So I asked him, like, what do you see from these, you know, how do you balance robustness and all these functionalities? And then you don't, basically, right? Sometimes you just have to figure out under what circumstances that will not fail. But that's another problem. If you put something in a very well-contained environment, that you probably have a better chance of like having it working. But again, you put them in human patients, you also don't know how they react, right? If it's like young patient, old patients that has a different system, and then pre-treated versus not treated, you know, with chemo, that also have a very different environment. So part of it for me is to define those variables. It's really hard to engineer anything if you don't understand the environment that you're working in and the substrate that you're working with, right? I think that's the main challenge. It's still going back to the basic science, define those environments. It's kind of like when you send a Mars rover to Mars, you kind of need to know what the Mars environment is before you can design your Mars rover to work in there. And we are still at that stage where we're trying to define our environment. So that's how I think about it. I don't have a solutions. You just end up trial and error, right? That's all I can say.
So that's interesting, Wilson, because I always think of the CAR-T cells as really the driver of mammalian synthetic biology, because they are the ones that made it to the clinic. So I'm just wondering what learning, you know, we can have from those CAR-T cells for the rest of us who are working with slightly different systems.
I get lucky. That's all I can. It was a lot of luck, I would say. I don't want to take any credit away from the giants in the field who made some amazing progress. But if you talk to them, there's a lot of luck involved. They chose CD19. It's worked remarkably well. You know, and then one of the early patients, Emily Whitehead, right? She had this massive fever. If you heard them talk, right? And then they used tocilizumab, IL-6 blocker, to help her survive. You know, if you ask Carl June, they really think that if she didn't survive, the whole field would have collapsed or have a big setback. So there's a lot of luck involved, and also like 20, 30 years of research in like T-cell engineering. Like you were saying, like engineering was important because long time they could not have a robust T-cell manufacturing process going. And that took 20 years, right, of like virus optimizations. And so I think, yeah, I don't know if there's any particular lesson, but at the end of the day, you got to see, it was one gene that they introduced. It's a one gene. So as a synthetic biologist, that's very not satisfying, right? I always want to do more. I always want to introduce a circuit in there. And right. And then all the clinicians looked at me and said, this is cute. Right? I get this look at, that, yeah, we actually want something like, it's not simpler. Can you like just give me another tiny little switch in there as well, like a drug inducible switch on top of that little switch that not little, but they're important, but that's, I always have to fight that urge of building something too complicated because that's what I want to do. But at the same time, the need is much simpler. Sorry, I digress. I don't know if I say luck is luck and lots of research.
I think that's a very interesting point, Wilson. And I suppose there's luck at many points in this in the progress from research, right? And I can imagine that luck will always play some part in the fundamental discovery, just deciding what to do. But it does seem worrying that the luck of whether one patient did or didn't die, I mean, bless her, I'm glad she survived, but the idea that one person dying or not dying can define an entire field and, billions ultimately of investment does seem a bit worrying that that's the kind of knife edge research is balanced on. So building on that and thinking about people's interest in going to the clinic, in 50 years or 20 years time, do you think we'll still have the same kind of approach of, test tube to a mouse to a human? Will we do everything in organoids on a chip, or will we just have a computer which knows what's going to happen and we don't have to test anything? What's your your dreams, or at least your realistic dreams?
Maybe to start with, we will test things directly with the patient cells. Hopefully things will have been accelerated this much that rather than testing in models, or we can just do the testing directly as personalized medicine as well.
Or what I hope that we can even eventually in the future bypass the patient's cells, like we have, there's a lot of talking about the off-the-shelf cells that you can use. So eventually, hopefully, we will be able to use something that is not rejected, that is not, I don't know how far in the future this will come.
Yeah, I would say in the field of cell therapy, to Wilson's point, understanding your substrate, right? I imagine, 50 more years of fundamental research is going to really help a lot, and that'll make it more like a test tube with 35 enzymes like what we work with.
I think to some extent, understanding the human physiology, of course, in my work is the immune system, right, as a system rather than as a cell or like few cells, right? It's how it interact with the heart, with the brain, right? And all these things matters. I mean, we've seen that kind of like organ-based modeling before, right? I mean, like like each organ is like a unit operations, right? We've seen that worked, fairly well actually. But when we drill down to the molecular level, to cellular level, of course, the complexity exploded and we couldn't handle those kind of modeling yet. I'm hopeful, but it requires a lot of like international large-scale collaboration to really define the cells, the organ, the tissues, and then the whole organism modeling. And it has to be human. I'm not a fan of animal model because I always get complained, no matter what I do, I'm wrong. So I would like to transition to organoid, but of course that also missing a lot of things, right? And yeah, but if as much as possible, I really don't, especially when I do like immunotherapy, like human immunotherapy, I have to use human cells. I cannot test, it's not a compound where I can test it on a mouse and then I can have reasonable expectation they might do the similar thing in patient, right? Because at least the drug product is the same. I don't have a mouse drug product and a human drug product Whereas with CAR T, with RT cell therapy, I'm using a human drug product, and it's different. Right? I don't want to develop a therapy for mouse because, as far as I know, they don't have good insurance, so they don't pay. So so I really don't. I don't really want to work on mouse as much as I can, but I'm looking forward to these major collaborative effort to help us define the human body.
If I can, sorry, I wanted to echo what has been said about the foundational research, in the sense that some time ago, we wanted to start a project on building some switches responding to tumor stimuli on immune cells in the tumor microenvironment. But then the specific thing I was interested in is so unknown still, and there's still so little known about how the specific biomarker coming from the tumor cells makes the immune cells respond, and the overall cell response, not just a single pathway, but all the interactions within the cells, that then we diverted into proof of concept thing, where you use the synthetic receptors, you use the synthetic molecules, but that is very far from being the actual thing. So I agree that also a lot of research and knowledge is needed.
Do you think this knowledge will be driven by academia, or are we going to be outgrown by the massive biotechs which have been cropping up, and I think things like ginkgo and other ones which are coming up more into the therapeutic space?
I mean,
how can we stay relevant?
I generally like like in communal effort. I don't like to see like enemies or divisions. So I hope that together we could grow into the knowledge, because everyone will have a different approach. And I think being a synthetic biologist means that you're open to interdisciplinarity and the difference in the way we look at things. So hopefully that will be coming all together.
Keith, perhaps you have something to say on this, given your involvement in various startups, right? Do you think it's going to come, all this innovation, is it going to come from small groups, grassroots, academia, startups, or will it be massive billion-dollar budget companies who have to ultimately be the ones to drive the whole product?
I think, yeah, I agree with Francesca. I think we need both. We need the resources of the larger groups, but Ginkgo Bioworks did evolve out of academic settings. So, I think there's also like the factor of time, right? If we fast forward 10 years from now, there's probably going to be a lot of the struggling startups today may be, that next big thing. So I think, I think basically we just keep doing what we do, and some of the things will work and others won't, right? And that's okay. Yeah, but I think, we just keep going. I think it's working.
Right, the last point I wanted to raise before we go to some audience questions was one about, I guess, building on this to make sure that the things invented, whether by academia or companies, are able to be accessed by as many people as we can, make as much benefit as we can. As Wilson said, ultimately someone has to pay the bills, and so often these things are being developed initially in large markets like the US, where there's a massive biotech funding available. Is there things that we can do in academia to make sure as much of this ends up in the hands of those that really need it? I mean, a common one recently, perhaps you've seen this a lot, Keith, as well, with your COVID work, is that, look at the vaccine rollouts. The AstraZeneca one has been doing a great job in the developing world because of kind of trivial things like the fact that there's not enough -80 fridges to go around. That on one hand seems like a bit of a small problem for humanity to face, but in reality, these are the kind of problems faced every day by billions of people. Is there anything we can do to try and improve the chances of getting as much impact as possible from our work?
I think that's like a huge part of what our lab is focused on, is this sort of improving accessibility, right? Like you said, there's a huge global population that lives outside of sort of the daily access that we might have in Oxford or Toronto or Boston to healthcare. And I think biology is this really interesting platform where if you have carbon, hydrogen, oxygen, those like you can build things in a very distributed way. You can decentralize manufacturing. And so I think, you know, as these technologies roll out of the lab, they're going to be extremely expensive. But over time, I think there's real opportunity for biology, because it's biology, self-replicating technologies. I think there's a real opportunity for bringing costs down, improving access. Yeah.
Elise and Francesca, you've both highlighted the difficulties of working with, say, mammalian cells. Is it conceivable that we could have these grown at the point of care in a place where there's not necessarily a massive biotech factory chemical plant for them? Is that ever going to happen, or is it always going to be, centralized manufacturing and shipped to end user?
It's technologically challenging. It's costly, and it's become, hopefully, as Francesca said, it will still be off the shelf. But I can't really see this happening in the near future. I don't know, what do you think, Francesca?
No, yeah, not in the near future for my experience. I mean, of course, it's limited experience, but ideally, you would be able to have small bioreactors, small production at the point of care. This is all nice to say, but then technically it's very challenging. And I think don't want to say things that are obvious, but basically I will say it, because it's I think it comes also from a rearrangement of the funding of the research, the accessibility that specific labs in developing countries have, towards the technology that we use. I mean, SynBio is mainly West-based, right? And so I think it takes time to change the governance of these kind of things. And I'm hopeful, as Wilson was saying, like I know that things will come, but it will take time.
I think it's West-based, but it's also very, we have a very enthusiastic younger generation that is a bit more international, and it's also coming from developing country or middle-income countries. And I think we can really use this base to sort of democratize a bit this knowledge that at the moment is within our hands, but could easily be transferred locally.
Yeah, no, it's true. If you think about SynBio Africa, so the IGEM like input into African universities, that is very nice. So I agree with Elise that there is enthusiasm and the will of growing.
But I'm not sure it will be enough for mammalian cells, though.
I also want to add, I don't think science will solve all of these problems. It also comes down to policies and incentives. I mean, you look at a lot of these small molecule drugs, it is, cost of manufacturing is pennies, and they charge you hundreds of thousands of dollars, right? Because they got to recoup all the investment that it's a business, right? And cell therapy, I think about these advanced cell therapy, they're expensive, but eventually the manufacturing will go down, the cost will go down. But then will they pass that cost to the consumer, right? Or is there any reason for them to pass the cost to the consumer, right? You are hoping that the market competition will do that, but it doesn't always have to be, right? Everybody just try to make as much money as possible, so they really don't have a lot of incentive to do that. And then it's just the same idea why no one developed antibiotics, because they can't make money. So there needs to be a different incentive, right? When you think about like these life-saving product, right? How do you incentivize it? Right, I think that's a big challenge. I don't think I can do the most awesome synthetic biology and then CAR T. Let's say I can make it for 1 penny each. It will still cost hundreds of thousands of dollars, because sadly that's what people can charge for that, and they will charge that much. So I don't know, right? How what I can do on that front. So So I agree with everyone, everybody says on the panel, but in addition to that, there's a need to be at a policy level, a global policy level that I'm just not smart enough to solve.
I don't think we can fault you for that. I think it's a global effort that's required. Yeah. Sorry, Keith, were you about to say something?
I completely agree. I think that's a big challenge, right? But I think there are efforts like the open bioeconomy at Cambridge, right, where they're trying to decentralize research so that research can occur in lots of, not just in a limited number of countries. And so, that's a great example of like, that's a science effort, but there's also a lot of other like components to that project that are helping enable it, like the legal frameworks that allow like open MTA and things like that. So it will take a long time, but eventually, maybe there will be generic CAR T. You never know, right? I don't know. But I think that's a really extreme example, right? I think there's a lot of other things like single molecule, like proteins, RNAs that are much lower cost and easier to distribute, to sort of, you can go to there's a lot of, like, for example, Brazil has a lot of or India, a lot of vaccine production, right? Locally, huge manufacturers in those therapies. And so, that type of, over time, these sort of cutting edge technologies will move to manufacturing in those settings,
I think you've both raised a lot of good points there. Building on this idea of the policy and financial challenges, undoubtedly the fundamental research costs a lot of money, but so too does the clinical trials, development, testing. A question in the Q&A box from Chris Finnis is, how does the panel believe regulatory approval processes need to evolve for these cell-based therapies? Is there, I mean, it's a different kind of ballpark to a drug, right? Putting a live cell into somebody. Do we have to change things there? Should we be changing them for the better as we get more ability to predict what might or might not happen? Could we make them cheaper and therefore make things more accessible? Or do you think we need actually more stringent requirements if we have more complex kind of human engineered medicines? Interested to see what anyone has to say on that end.
Yeah, I don't know if it's a regulatory issue so far. At least in the US, I'm surprised how friendly it is to the regulatory agency to sell therapy products, cell and gene therapy products. The last few years, we've seen a lot of approved products for CAR T therapy and a few more gene therapy. That's insane, right? You think about, there are 7 checkpoint inhibitors approved, 4 CAR T therapy approved in the last 3 years. I'm surprised, actually, quite friendly it is. But I think that at least the US regulatory agency is very willing to work with companies. But I think the challenge is at manufacturing, right? A lot of the issue comes in CMC. And mostly, again, because we don't know the substrate well. Like the virus production is very difficult, very expensive, very challenging, because we're using hex cells to make virus and they don't always make them clean enough. They spit out all these other impurities and you don't really know why. I don't know why. I think that's the big issue. And then patient sample comes in highly variable. So I don't know. I think at this point right here is a lot of them come back down to manufacturing, right? And that science can solve. I think the regulatory agency around the world is trying to work with companies and try to define what's a stringent. I think still a work in progress, as my understanding is. It's an evolving guideline. So yeah, that's what I want to say about the regulatory issue. I'm actually quite surprised how friendly it is.
As I was listening to Wilson, I was wondering, are those regulatory issues a bit more open when we're talking about therapies that are applied as a last resort? So is it part of maybe why, for the CAR T cell, as you say, you were surprised how easy they would be to be applied in the clinic, maybe because those are for conditions that have not much other alternatives. What about the rest? What about other conditions where we are trying to build up alternative possibilities? And then I'm wondering, how would the regulatory landscape be in those situations?
That's a good point. I think what CAR T does, it gives regulatory agency an example, right? When you say, well, if I introduce engineered CAR T cells into the patient, will they become malignant? Will they become cancerous? And now we have thousands of patients, hundreds of years of patient history saying that no, not a single one of them turned cancer cell. But that does give a lot of, right? That helps the other field, right? If you're going to engineer immune cell and say that, you don't worry as much, right? Or at least like if you do viral integrations, like now we say, well, if it didn't integrate in these places, they're unlikely to become cancerous. So now we have data to support these claims. So I think it helps too. Yes, I think of course in the beginning, a lot of these CAR T is like, well, this is a second line, third line, fourth line, fifth line, right? And I think for a lot of advanced product, it's always going to be like that, right? If you can take a small molecule just for the cost and convenience, that's what you will do a lot of the time. But I would say because of the success of CAR T, and even though CAR T is nice in a way that even though they have side effects, you do know what they are, right? It's not a surprise side effect. Almost every time comes up the same thing, right? So you do know what to look for, right? As opposed to the other one that you don't know when they break down the product, what are the side effects. So it's nice like that. And then it does give you more, it opens up to other cell therapy. I do say, I think so.
Yeah, it's a good point. And building on your manufacturing challenge, another question we have from Brian Andrews is, do you think there's a significant role for the open source approach? I mean, I have a slight horse in this race in that I run an open source bioreactor venture. But others, we've mentioned Cambridge and their open bioeconomy. Do you think these technologies are, I mean, I'm personally quite skeptical that my bioreactors will ever be useful for building like useful amounts of a chemical. Do we think the open source is the answer to try and get this stuff more widely used, or will it potentially lead to problems? Lots of people accessing engineering, doing fiddling. Can that create more harm than good? That's always been something I've heard a lot when I've been into this open source debate with other people.
I think basically the open source today is an excellent sort of venue for research, right? Like it's going to expand the number of people that can do research, which is going to, more the Canadian sort of saying more shots on goal. And so I think it's a great thing. I think being able to do open source therapeutics, or that, I think that's harder. I think there's, like we've been talking, it's expensive, and you need incentives to take, to incur all of that expense. And so I think that's going to be a harder one. That maybe if there's like sort of national consortia that come together to do things that are really critical, maybe. But for now, I think open source is great for research, and it'll spawn lots of new technologies.
I want to add to that a little bit, at least in the CAR T and the cell therapy. What they say is the process is your product. So it's everything. It's not just, you know, the car molecule, right? It's how you make it all the way from the beginning to the end. That's what you're selling. That's the whole process. And you can't change from it once you get approved, right? Once you run that pivotal trial, you can't do that. I'm going to change to something else. That's not going to fly. Right? You need a different approval process. So I guess in the very beginning, let's say there's a, I don't know, there's a promoter coming from the open source that worked really well. I don't see why it cannot be incorporated into your development in that way. But at the end, it's the whole process, right? What buffer you use, you know, what flask you grow your hex cells with. That you cannot change. So yeah. So I don't know. I mean, I think there's something you can definitely take from the open source. I feel like, but not everything else is locked in from the regulatory standpoint.
Francesca, sorry, go ahead, Elise.
I really like the idea of an open source bioreactor. I think there's a lot of recombinant molecules that are easy to produce nowadays, and that are life-saving, and that could, you know, with a bioreactor could be produced fairly easily. I'm thinking insulin here. And I think the problem are not only production, it's everything that's downstream. So it's purification, distribution, also how you deliver it. So I know that we were looking at insulin distribution in low-income countries where some people still don't have access to this life-saving drug. And it's not only due to the price or even the accessibility to insulin, it's also accessibility to the knowledge, the syringes, etc. So I think it needs a bit like Wilson was saying, if you're open source, you need the whole of the approach to be looked at.
Yeah, that's certainly a very good point, and I think it will be very challenging. And there's an interesting group in, I think, San Francisco called Open Insulin who are trying to do this using all open source, and I've helped them in a few points, but it does seem like there are some formidable challenges there at many steps of the pipeline. Anyway, just trying to get through some more of the audience's questions. So we have a question from Wabnik, which I might direct to Francesca because she has worked in several different host organisms across her career. And that is, do you think it might be easier to, instead of necessarily engineering mammalian cells directly, maybe we could engineer microbes? I'm thinking things like microbiome, gut microbiome, to have them then interact with the mammalian cells to implement therapeutic functions? Is that going to be easier, or is it more challenging?
It's a very good question. I think probably Elise is more experienced, and this is actually your topic, right? So, I mean, that's definitely an option, especially depending on the, if you think about the microbiome, right, and the human microbiome, the gut applications. So, of course, using microbes themselves is way smarter if you can biocontain them and you know, make them safe. So I would say, of course, it's an option. Yeah, I don't necessarily have a lot of experience in bacteria mammalian interaction and projects. So
I think we still need to learn a lot. We're still in the learning stage again in those interactions, but definitely having bacteria as chassis seems a bit like a lower hanging fruit to me. Technically, cost-wise, how much easier, you know, development, optimization, testing would be. Then, so if there are ways that, you know, those systems could be engineered to work together, I think it would be a nice way. But unfortunately, it can't always be the case. We still need to engineer the mammalian cells.
I think we just have 1 minute till time, so maybe I'll ask just in the last couple of minutes each candidate, maybe 30 seconds, just closing remarks. Tell us your most exciting thing coming up in therapeutics in bio? What should all the students in the audience, or maybe undergrads, even high schoolers, what should they keep their eye on for their future career and think about getting into? Do I nominate someone to go first, or is someone feeling brave?
I
Maybe
mean,
Francesca.
I look with a lot of interest and hope to do something in the topic. When I look at the Human Genome Project, right? So that is something that it's really interesting. And it's I think that it's going to be, of course, it's going to take time, but if we can improve, as we were saying during the conversation, our ability to engineer mammalian cells in a more knowledge base. So the idea of the project is to try to understand the mammalian cells much better and develop the tools that we are currently lacking for doing this engineering, right? In a way that is much faster and easier. Don't know when, but then this is something that I think can hold promise for the future, for sure.
I like, so if I'm allowed to go a bit sci-fi wise, well, actually it was really sci-fi 5 years ago, it's not so much now. I like the xenotransplantation approach. There's still a lot to be done there, but I think it's very exciting.
I guess my only advice would be that if you're sort of a student in this space, that no one really knows what they're doing, so you can't really make mistakes. And I just say go for it, and don't worry about that. So that would be my advice.
Yeah, I'm. You can tell I'm a big fan of immune engineering. And I'm also actually, at least I'm a big fan of xenotransplant. I think that will solve a huge number of problems. Like, for example, you have lung cancer, right? Right now you try to kill the lung cancer cell, but if xenotransplant work, I'll get a new pig lung instead, right? It's a different way of treating diseases. But these things requires also understanding and reprogramming the immune response as well. So I think there's a big opportunity for, in terms of engineering human cells, I would say immune cell engineering is actually quite more accessible, right? It's hard to engineer a kidney, right? You can't take it out and then do something to it, put it back. Whereas immune cells, you can. So that's the nice thing. That's sort of like the cheating way. I didn't recognize it before. Now I'm like, okay, this is really just cheating because I don't have to worry about like specificity and delivery and all these things, right? So I'm a big fan of that. You can do a lot and you engineer the immune system. I think it's reprogramming the entire immune system, not just one cell type. That's what my sci-fi envision, and that can have impact, of course, in cancer, infectious disease. What I really want, and healing as well, like how do you heal, like can you use the immune system so that it heals faster, like Wolverine? I think there's a way to do that. Perfect healing, no scars, right? So that's awesome. Heart disease, people are trying that. I think there's also a chance where you can engineer the immune system to influence psychological behavior as well, because they talk. This neuroinflammation is bidirectional. So it's hard to engineer the brain. But I think there's a handle where you can engineer the immune system to influence like post-traumatic stress disorders, right? And things like that.
I hope we'll also get the Wolverine claws while you're being so generous with our new features, Wilson. Anyway, this just about brings us to the end of the academic panel. So thank you very much to everyone, to Wilson, Francesca, Elise, Keith. Thank you for joining us today and talking to the Society. It was really good to see everyone's face, even if we can't do it in person, but hopefully at a conference someday soon. So now we have just a couple of minutes. Everyone can grab a break before the next session.
Thank you.
Thank you all.
Thank you. Thank you.
Thank you, everyone.
Hi,
Since
Corinne.
the panel discussion ran a little bit over, we don't have much of a break. I think we'll go right into the startup presentations with our chair, Jose.